So high performance liquid chromatography, HPLC. Now, as the name implies, high performance liquid chromatography. Now, this is a chromatographic technique. So it works based on the principle of other chromatographic techniques. That is, all chromatographic techniques have two phases, right? Mobile phase and what? Stationary phase. Mobile phase and stationary phase. So the high performance liquid chromatography still has mobile phase and stationary phase, but it is high performance because it works at high speed. Now it is very similar to column chromatography, but here we apply pressure, we apply force so that the separation will be very fast. Now we have four main types of HPLC. So the four main types are normal phase, reverse phase, size exclusion, and ion exchange HPLC. All right, so let's look at the first one. Now we've seen that in chromatography we have stationary phase and mobile phase, right? Now, in the normal phase HPLC, which is the normal one, the stationary phase is polar, while the mobile phase is non-polar. Now, when we say something is polar, it has partially positive charge and partially negative charge. Now, normal phase, the stationary phase is polar, while the mobile phase is what? Is non-polar. So it works based on polarity. So it separates molecules based on polarity. Now, let me ask, do you think this should be used to separate polar substances or non-polar substances? Will it be used to separate polar substances or non-polar substances? It will be used to separate polar substances, right? Because if you are using it to separate non-polar substance, remember the solvent here, the mobile phase here is non-polar, right? So if you are using it to separate non-polar, it will be interacting with the non-polar because non-polar interacts with non-polar. Polar interacts with polar. Now, this works mainly based on polarity. So it separates molecules based on what? Polarity. What you have to note in this normal phase is that the stationary phase is what? Polar. And the mobile phase is what? Non-polar. That's that. Then in the reverse phase, reverse, meaning you are turning the normal phase, is it not? In reverse phase now, the stationary phase here is now what? Is non-polar. While the mobile phase is what? Polar. That's the underlined thing there. In normal phase, stationary phase is polar, mobile phase is non-polar. But in reverse phase, the stationary phase is non-polar and the mobile phase is what? Polar. That's that. Then size exclusion HPLC. In size exclusion HPLC, it separates molecules based only on what? Size. Based only on the size of the molecule. So take for example, you have something like this. Let me use this as our stationary phase. And these are pores, these are holes, so small holes. And you pour your mixture you want to separate there. Add your pressure. You'll notice that the only things that can pass through these small holes are molecules that are smaller, particles that are smaller than the holes, right? It's just like you're sieving gari, not drinking. Like when you're preparing gari, that time you put the gari on the mesh to sieve. I think you notice that after sieving, some things will remain on. People that have not seen cassava processing before will not understand this one. After sieving, shaft will remain on top, right? Those things were not small enough to pass through the mesh. That is like size exclusion. So you are excluding some based on size. So in size exclusion HPLC, you are separating molecules based only on the size of the particles. Do you understand that? In ion exchange HPLC, you are separating the molecules based on what? Based on what? Based on charge. Based on charge. So, you know, we have, when we talk about ions, we have cations and what? And anions. Among cations and anions, which one is positively charged? This one is what? Positive, right? Why this one is what? Don't forget this plus here in the cation. So it makes it positively charged. And this anions make you cry. Negative. Alright, so this is it. Now we want to separate cations from anions. Now check this. If I have something like this and the column is made of positive charges, let's say the column is made of positive charges, and I have a mixture of positive and negative charges in what I want to separate, what do you think will pass through the column? Positive, right? Because negative, this positive column will attract the negative. Is it not? We attract the negative, hold them captive. So only the positive will go out, because positive like charges repel, right? While unlike charges attract. So ion exchange HPLC separates based on charges. Size exclusion HPLC separates based only on what? Size. Reverse phase. The reverse phase, the stationary phase is what? Non-polar. While the mobile phase is what? Polar. While in normal phase, the stationary phase is what? Polar. And no, so this one separates based on polarity. This one separates based on what? That's hydrophobic interaction, right? This one size and this one charges. Do you understand that? Alright, so let's look at the components of HPLC chromatogram. Now, when we talk about chromatogram, the one ending with M, we are looking at the instrument used. We are looking at the instruments used. So we want to look at the parts. If you are asked to write on the instrumentation of HPLC, this is what you give them. If you are asked to write the components of HPLC chromatogram, remember the chromatogram is talking about the instrument used, right? Good. Now, if you hear, alright, so that's that. If you hear chromatograph, the one ending with PH, chromatograph is the output, what is given out. Chromatogram is the instrument used. Now, the components of the HPLC chromatogram, number one here is what? HPLC pump. Remember, I told you HPLC, high what? High performance, right? Some books you see high pressure. Now, it's not allowing in HPLC, it's not like other chromatography that you just allow the separation to take place normally at normal speed. Here you want to hasten the process. You want to hasten the process. So you add your own pressure, you apply pressure. Now the HPLC pump, yeah, is what is giving that pressure to pump the mobile phase. The mobile phase is that liquid, that solvent where you add the analyte. The analyte is what you want to separate. So normally you pour your sample. Is the mobile phase that carries the analyte through the column. But now the HPLC pump is what is providing the what? The pressure that will carry the mobile phase throughout the HPLC system. Do we get that? So it is the HPLC pump that is giving that pressure. And the HPLC pump we can have pneumatic pump, we can have syringe pump. How many of you you've seen syringe, right? It's just like if you put injection in that syringe. Injection is a liquid. It's just like you put injection in the syringe and allow it to be dropping like this. Dropping. And when you push that plunger, which one will be faster? Pushing the plunger, right? Good. That you are applying pressure. Exactly what the HPLC pump is doing. So the HPLC pump provides the pressure that will carry the mobile phase, the injection now, throughout the system. Do we get that? That's the HPLC pump. Now the HPLC mobile phase is a solvent. Remember that chromatography, you have two phases, mobile phase and what? Stationary phase. The mobile phase is the HPLC solvent here. And what is the HPLC solvent doing? It is carrying, it's just like, now look at this. In this pump here, sorry, in this column, it is very dry. And you want to separate something, you just put the substance you want to separate there in that dry. It's not even move. So we need a solvent that will carry the sample throughout the column. So what the mobile phase is doing is to carry the solvent, is to carry the analyte, the mixture throughout the column. If you forget what I've said so far, don't forget that. HPLC pump provides the what? The pressure needed, right? Mobile phase carries the what? Analytes. Is it not? So the mobile phase is the solvent we are using here. Now, the HPLC injector, let me use this. Now, you pour your solvent, that is the mobile phase, in your HPLC pump. Now, as you pour your solvent in this HPLC pump, what will the HPLC pump do? Push that solvent with high pressure, right? Apply pressure, push it. Then here, we have something here called the what? Injector. The injector here, the HPLC injector, is where you put your sample, where you add that mixture that you want to separate. Do you get that? So the injector is used to inject the sample into the column for separation. Do we follow? Are we following? The sample is added into the injector now injects the sample into the column. Then as you inject the sample here, remember that mobile phase is coming from here with high pressure, right? The mobile phase that is coming from here with high pressure will now carry that sample throughout the column with high speed. Now here we have the column. The HPLC column is made of stainless steel. We've seen stainless steel, right? You've seen that stainless plate. Something like that. Stainless steel. And it is packed with silica gel to create that stationary phase, because you know that separation is done by interaction of the sample, components of the sample with the stationary phase, right? Now, the column here, this the column, and you know the column is having as a stationary phase, is it not? Now, here you pack this stationary phase with silica gel. So these are stationary phase. Your solvent is added here. This one pumps it with high pressure. The injector is where you put your sample. So now the mobile phase carries the sample through the column at high speed. And as they pass through this column, separation will take place, right? Once one sample is one component of the mixture is separated, that one will be eluted. And we have something to detect it here. As another one is separated, that one will be eluted. You have something to detect that another one is here. Just like that, because they will be eluted at different rates based on how they interacted with the stationary phase. Then we have something that we analyze that. Alright, so the HPLC degasser, if you use the prefix D means what? Removal, right? Degasser means removal of gas. HPLC degasser is used to remove gas from the mobile phase. So it's something that is used to remove gas from the mobile phase. That's the HPLC degasser. Alright, I didn't mention that the HPLC injector can be of Rheodyne injector, septum injector, stop flow injector. These are the different injectors that can be used. And I said degasser is used to remove gas from what? The mobile phase, right? Or the solvent. Now, HPLC oven is used to regulate the temperature of the column. Is used to regulate the temperature. HPLC detector, we said, is used to detect the components that have been eluted, right? Is used to detect the components that have been eluted. Then we have our the HPLC detector can be of visible light detector, UV detector. We can have our mass detector, fluorescence detector, and what? And what? I gave you 4. Infrared detectors, right? So this is the components of HPLC chromatogram. So if you are asked, please ensure you draw this. I've told you this is instrumentation course. So to draw this now, if you notice, I did not represent all the part. I did not show the oven here. What else? The degasser, because they are not key components of this. But the key components, this is the mobile phase, right? The mobile phase, you put it into the pump. The pump will now carry it with high pressure through the column, right? But we have where we put our sample, and that place we put our sample is what? Injector. So now the pump will now carry it through the column, which is our stationary phase. It will now interact with the stationary phase and be eluted. The components will now be eluted one after the other. So we have a detector to detect that. Let's look up applications of HPLC. Now we've seen that HPLC is used in separation, right? So we're actually separating, but now we are separating under high pressure. Now we said that HPLC can be used in pharmaceutical industries, and in the pharmaceutical industry we can use HPLC to monitor drug stability, how stable a particular drug is. We can use it to analyze tablet dissolution, how tablets dissolve. That's what we're talking about, a tablet dissolution. We can use it to check the general, we can use it for general quality control. So in pharmaceutical industry, you can apply HPLC for these purposes: monitoring drug stability, tablet dissolution analysis, general quality control. In environmental biochemistry or environmentally, we can use HPLC to analyze or to check for pollutants. Apart from that, you say you can also use it to check for what? Environmentally. Apart from pollutants, what can you use HPLC for in the environment? Detection of enolic compounds, right? Where? In drinking water. In fact, generally, you can use HPLC to analyze drinking water. You can use it to analyze drinking water. Now, in forensic science, forensic, we are talking about crime detection, is it not? In forensic, you can use it to detect the presence of particular drug. You can use it to quantify drug, the quantity of drug in the human body. Forensic. Clinically, we said in clinical practice, you can use HPLC to analyze what? To analyze what? Urine, presence of urine in the blood, presence of bilirubin in the blood, antibiotics. So you can use HPLC to analyze the presence of all those things in the blood. Bilirubin is gotten from bile, right? Yes. And bile is gotten from the breakdown of red blood cells. If you break down red blood cell, what you have is bile. If you break down white blood cell, what do you have? Pus. Anytime you have injury and pus starts coming out, no, that's the first sign of infection. The white blood cell that was protecting you, fighting against, have been killed. So when white blood cell is killed, you have pus there. All right, that's by the way. Now, in food and beverage industry, you can use it to analyze what? Polycyclic compounds in vegetables. What else? Analysis of what? Preservatives and sugars. There are some preservatives that after they've analyzed them and checked their effect, they no longer use them. There is one that is used in bread, potassium bromate. They banned potassium bromate. They no longer use it in bread because of the effects. Say it have been found to have carcinogenic effects. So that is why some bread now, you see they write on the body bromate free bread, because they've banned potassium bromate because it has been found to be carcinogenic. So in food industry, you use HPLC to check most of those things. So basically, these are some of the applications of HPLC. So looking at the advantages of HPLC. Number one, it is simple. HPLC is very simple. It is automated in that once you start the process, you are not in control of the intermediates. So you just start the process and it goes on. So the process is automated. It is simple, automated, and highly accurate compared to, if you compare this to thin layer chromatography and paper chromatography, this is the most accurate. So it provides high resolution. So it is simple, automated, and very accurate. Number two, it can be used in quantitative and qualitative analysis. Now, note that when we talk about quantitative analysis, in quantitative analysis, we are looking at the amount of a particular substance present, right? But qualitative, we are looking at, is it present or absent? So qualitative, is it present or absent? Quantitative, in what amount is it if it is present? Do you understand that? So you can use HPLC to determine the presence or absence of a substance, and you can use it to determine the amount. So it's used in both qualitative and quantitative analysis. Now, it can be upgraded to mass spectroscopy. Paper chromatography cannot be upgraded to mass spectroscopy. As we go on, we'll be looking at, in our next topic, we'll be looking at spectroscopy, so you know what it means in details. But just for now, just note that HPLC can be upgraded to mass spectroscopy. And it is faster. Since we are applying pressure, it will be very fast, and it is effective also. So these are the advantages. But you know that anything that have advantages have what? Disadvantages, right? Now the disadvantage now is the solvent, which is the mobile phase, is very expensive. The mobile phase is very expensive, and the procedure requires steady power supply. So if you don't have steady power supply, you cannot still carry out this process. If you don't have money to buy the solvent, the mobile phase, you cannot carry it out. Number two, it needs an expert. So you must have idea of if they just take you now to where this HPLC is and say, oh yeah, this is the sample we want to separate, oh yeah, start. You'll just be looking at the machine like this. So it requires an expert in the manipulation of the equipment. Contaminated column can affect peak shapes. Now, remember that the detector used have the ability to convert electrical signals into peak like this. And it is this peak that can be used to tell the quantity of a particular analyte. Now, if the column we're using, the HPLC column, is contaminated, the shape of this peak will be affected, and so we'll have wrong result, false result. So that is why cleanliness of the system is required. Alright, so these are the advantages and disadvantages of the HPLC. Any questions so far?